Hello, my name is Yuliana Guerrero.
I'm Marcus Pitts.
I'm Carson O'Pry.
And I'm Esther Oyekan.
And today we will be talking about atrial septal defect and the role of GATA4 and application of CRISPR. Atrial septal defect, also known as ASD, is a congenital heart defect where there is a hole in the heart that separates the two upper chambers of the heart. It is by far one of the most common defects. Despite its prevalence and clinical significance, the cause of ASD is unknown. GATA4 mutation is mapped to a region of chromosome 8 where deletions and have variable associations with ASD. To further test this, we suggest working on pigs due to pigs expressing genetic and psychological traits like humans. We propose using the CRISPR to turn off the mutated gene in pig embryos to better understand atrial septal defect and the root cause of the defect. And now I'll be passing it on to Esther.
Hi. So ASD cause is unknown, but it has been associated with GATA4 mutation. Congenital heart defect is more commonly diagnosed in patients in developed countries who have higher incomes. Some symptoms of ASD include chronic lung infection, trouble breathing, shortness of breath, arrhythmia, heart murmur, stroke, swelling of the legs and feet, and the stomach. Most young adults with ASD present with heart murmur or abnormal electrocardiogram. In adulthood, patients develop breathlessness or palpitation. Most people think ASD is born with the heart with the hole in the heart, but it is more than that. And most people think they don't live long when they hear about ASD, but a lot of people can live up to at least 50 to 80 years. And it is said that several patients with ASD can tolerate unrepaired heart for 80 years or longer without serious effect. The prognosis can vary from patient to patient depending on the severity of the defect. And now I'll be passing it to Carson.
Right, so there's some key concepts that we will be discussing today. One is about the GATA4 gene, which is a transcription factor that plays an essential role in heart development. The GATA4 gene is expressed in the oocyte and it regulates genes involved in embryogenesis and in myocardial differentiation and function. Also, we'll be discussing about CRISPR, which is a laboratory tool used to change or edit pieces of a cell's DNA. The CRISPR-Cas9 tool uses a specially designed RNA molecule to guide an enzyme called Cas9 to a specific sequence of DNA. The Cas9 then cuts the strands of DNA at that point and removes a small piece, causing a gap in the DNA where a new piece of DNA can be added. And now I'll pass it to Marcus.
There's been research already in GATA4 gene using zebrafish, house mice, and even sea squirts, but there's not enough research in pigs, and we hypothesize using the loss of function method to the specific gene GATA4 in pigs with hereditary ASD will end the heritability of the mutated GATA4 gene in the next generation. Funding our research will help advance the understanding of this gene and hopefully lead to breakthroughs in either finding a cure for ASD, preventing ASD, or just stopping it from being passed down. Each step of research will help the progress in this, and we hope to build the knowledge base and infrastructure that supports these efforts. And understanding these diseases in any life form will help crack the code for the human ASD. And using subjects with organs and proteins like ours, like pigs, can help identify the key players in congenital heart disease mechanisms. Thank you very much. That is all from us.